proliferation, they need many things. They need many components. They need many proteins, many other lipids, many components of DNA, many components of RNA. So they need another metabolism, cellular metabolism. You know, cancer cells and neoplastic cells demonstrate a distinctive form of cellular metabolism. They have a high level of glucose and glutamine uptake. They are glucose hungry. You know, the neoplastic malignant cells are glucose hungry. They show increased conversion of glucose to lactate or lactate fermentation via glycolytic pathway. You know, for producing ATP and for producing energy, there are some ways for metabolism, and there are some different pathways. One of them is glycolytic pathway, and the other one is oxidative phosphorylation. Okay. The cells in normal situation use oxidative phosphorylation because in this way they can produce many ATP by each molecules of glucose. But in the glycolytic pathway, they can produce fewer ATP molecules. But the tumoral cells prefer this way, prefer glycolytic pathway, even in the presence of ample oxygen. This pathway of aerobic glycolysis called Warburg effect. So, as I said before, the tumoral cells and also some rapidly dividing cells like inflammatory cells. For example, in the situation of infection and inflammation, the inflammatory cells are rapidly dividing cells. So, the tumoral cells and also rapidly dividing normal cells are glucose hungry, and they show Warburg effect. Warburg effect means that they prefer glycolytic oxidation and glycolytic pathway for producing energy. We can use from this feature in some scanning way. Because the cells are glucose hungry, we can use from PET scanning. What does it mean? It means that this patient, when injected by a type of glucose, this glucose taken up into the tumoral cells and also into some rapidly dividing cells. And after the injection of this type of glucose, we can find this area as PET positive area. You know, in PET scanning, we have some scanning by some hot spot in the body. Okay, we call these spots that they are hot. We call this area PET positive area. Some tumors, and most of the tumors are PET positive, and also some other area like some lymph node, maybe in normal situation, be PET positive. So we can use from this feature of the tumoral cells for finding them, for find the site, the exact site of tumor, for example, and sometimes for finding the metastasis site. Okay, for example, when we have a cancerous patient, a patient, for example, with prostate cancer. In the next step, we should use PET scanning, and by this scanning, we can find the sites of metastasis, and we can use relevant way for therapy and relevant chemotherapy for these patients. As I said before, in aerobic phosphorylation, which is used by normal cells in normal situation, can produce 36 ATP molecule per each glucose. But in glycolysis, which we said the tumoral cells prefer this way, only they produce only 2 ATP per each molecule of glucose. Okay, now the question is, why is it advantageous for a cancer cell to rely on inefficient glycolysis? I said before, the rapidly proliferating healthy cell rely on aerobic glycolysis. So Warburg metabolism is not only for cancers. As I said before, some rapidly dividing cells use this way. Okay, as you know, a growing cell has a strict biosynthetic requirement. They need DNA component, RNA component, lipids, proteins, and many other organelles, okay? So for any dividing and for proliferation, the cells need all of them. While pure oxidative phosphorylation yields abundant ATP, but it fails to produce any carbon component that can be used to build the cellular component needed for growth, as I said before, like protein, lipids, and nucleic acid. So, in the oxidative phosphorylation, we have only many ATP, but we have no this other component. So the answer is here. Aerobic glycolysis provide rapidly dividing tumoral cells with metabolic intermediates that are needed for synthesis of cellular components, whereas mitochondrial oxidative phosphorylation doesn't. So this is the meaning of Warburg effect. So how is this reprogramming of metabolism, the Warburg effect, triggered in proliferating normal and malignant cells, and how does it get hardwired with cancer cells? As I said before, mutations are the heart of malignancy. So again, in this altered cellular metabolism and in this difference in the cellular metabolism, we have some other mutations. So metabolic reprogramming is produced by signaling cascade downstream of growth factor receptors, the very same pathway that are deregulated by some mutation in oncogenes and also tumor suppressor genes in the cancer. And whereas aerobic glycolysis is in normal cells when they are no longer growing, but in cancer cells, this reprogramming persists due to the action of oncogenes and also loss of tumor suppressor genes activity. There are two other links between metabolism and cancer. One of them is autophagia and the other one is oncometabolism. Oncometabolism is a new entity and some new enzymes and new molecules found in the metabolism of the tumors, and now we have some factors for the chemotherapy and for making some targeted therapy for them. Do you know anything about autophagia? What's the meaning of autophagia?
Self-digestion of cells.
Why?
Due to stress, so they digest themselves inside the organelles.
Yes, which kinds of stress, for example?
I think malnutrition.
Yes, loss of nutrition, yes, exactly. So now about autophagia. Autophagia is a state of severe nutrient deficiency. Cells not only arrest their growth in this situation, and they consume their own organelles, proteins, and membrane as a carbon source for energy production. If this adaptation fails, after that the cell dies. Tumor cells often seem to be able to grow under marginal environmental conditions without triggering autophagia, some tumors, suggesting that the pathway that induce autophagia are disrupted in the tumoral cells. So we have some mutations that they can disrupt the pathway of autophagia in some tumoral cells. So genes that promote autophagia have tumor suppressive activity. They act like a tumor suppressor gene. Now, the question is whether autophagia autophagia is always bad for tumor. And I said that some tumors stop this process. Okay, in a condition of severe nutrient deprivation, tumor cells may use autophagia to become dormant. Okay, so some tumor have this ability and they can use autophagia to become dormant. This is a state of metabolic hibernation that allows cell to survive for a long period of time. Okay. And these cells, now we know that they are resistant to therapy in this dormant phase by this hibernation. And they are resistant to therapy that kill actively dividing cells. You know, many of the chemotherapy or radiotherapy can kill the cells which are in dividing states, the cells that they are in dividing process. So the dormant cells can be resistant to the chemotherapy and radiotherapy, and after stopping the drugs and the therapy base, after that we have relapse and we have recurrence of the tumor. Now about oncometabolisms. Another surprising group of genetic alteration are mutation in enzymes that participate in Krebs cycle. I think you know about this cycle. We have in many tumors mutation in one enzyme. We call them isocitrate dehydrogenase or IDH. Now this is very important and we can use them by immunohistochemistry. Immunohistochemistry is a way that we can use in practice in our lab. As I said before, this is a type of staining way by using the action between antibody and some antigen by using and attaching some stains and finding the type of tumor, for example. And now we have this marker. Now we have IDH for finding some tumor and for finding these mutations. Mutations in isocitrate dehydrogenase or IDH are of particular interest as they have revealed a new mechanism of oncogenesis, termed oncometabolisms, and they can use some drugs, some targeted therapy that they can target this enzyme and this process of oncometabolism. Steps in the oncogenic pathway involving IDH. At first, we have mutation in IDH. After that, we have some specific amino acid substitution, and then we have mutated protein losses. Exactly, we have loss in the ability of the mutated proteins, and then we have a neoenzymatic activity in some tumoral cells. And finally, we have production of another protein, 2-HG or 2-hydroglutarate. 2-HG acts as an inhibitor of other enzymes that are members of TET family. Loss of TET activity leads to abnormal pattern of DNA methylation. And finally, we have aberrant expression of the cancer genes. We have aberrant expression of the other genes like P53, like RB, like MYC. So these steps have many details, and I want you to understand only about that all of these steps and all of these are for some mutations in some enzymes and important molecules, for example, in Krebs cycle. And finally, all of them add to each other. And finally, we have mutations in the important cancer genes, you know, for example, P53, RB, MYC. These are the important cancer genes. Mutated IDH act as an oncoprotein by producing 2HG, a prototypical oncometabolite. Since the mutated IDH proteins have an altered structures, it has been possible to develop some drug that inhibits mutated IDH and not the normal IDH enzyme. These drugs are now approved for treatment of some certain cancer that they show IDH mutation, like acute myeloid leukemia. And
could we know the difference between the normal and the muted IDH?
Yes. As I said before, by some molecular study, we can find the difference between them.
No, I mean generally, what's it? Not how to detect the differences between them. Like generally, what's the difference? Why is that normal?
Oh,
Why
yes.
is that?
Any normal enzyme and any normal protein have an exact activity, for example, in the Krebs
Is
cycle.
it related to its activation and disactivation?
Yes. By mutation and disactivation of the normal IDH, finally, I said we have some altered protein, and after that we have mutations in other enzymes and in other important cancer genes.
So can we consider the mutate is activated and the normal is unactivated? Or no?
No, we can say that this IDH have a normal activity, not only the activation or not activate or inactivation.
And the mutate have
Yes.
mutate activity.
Yes. And there are some other important mutations, some important carcinoma that we can find IDH mutations in them, okay? For example, in cholangiocarcinoma, glioma, as I said before, acute myeloid leukemia, and many types of sarcoma, they can find the IDH mutation. So after finding this mutation, we can use the drugs that can make better the function of IDH. Cholangiocarcinoma is a tumor of, what's the meaning of cholangiocarcinoma? A biliary tract system. Yes, from the bile duct. Cholangiocarcinoma. This is a epithelial malignant tumor from the epithelium of biliary tract system. And about glioma.
Cells in the brain or spinal cord?
Glial cells, yes, exactly. Acute myeloid leukemia from blood cells, a type of myeloid origin of blood, and any kind of sarcoma. Sarcoma means malignant mesenchymal.
Is the glioma the responsible for reducing the brain tumor and something else?
Glioma. Yes. In glioma we have some glioma. We have two groups of glioma. Some of them are IDH mutant and some of them are not. Okay.
Related to malignant or not?
No. Glioma is malignant. Glioma is malignant. Yes. This is one of the other exceptions. OMA but malignant.
Yeah.
Yes, glioma are malignant.
What about sometimes we, for example, had some cases like it's a brain tumor, for example, but it's not malignant. So, what causes that?
Yes. For example, some types of tumor in any parts of the body, only we have proliferation in this area and we have cured them only by removing that, okay? And they don't have bad behavior, they don't have invasion surrounding tissue, they don't have metastasis, and they are benign. But most of the tumors by these mutations are malignant. And about glioma, we have some IDH mutated glioma and some non-mutated glioma. We should find them by practice and by some molecular way and also by some immunohistochemistry way. We should find them for using the specific drugs. Okay, now we should start the other class.
Do you suggest to study it like this? when we reach that point in that file, do you suggest to come and set this file, then we will complete it? That?
No, no, no.
Oh, it's fine.
It's fine. That's okay. Yes. Okay. Now about another feature of the malignant cell is development of sustained angiogenesis. What's the meaning of angiogenesis?
Formation of blood vessels of like carcinoma.
Yes, yes. Genesis means making anything. Angiogenesis, yes, making vessels. Yes. Even with all the growth advantages, tumor cannot enlarge more than about 2 millimeter in diameter unless they are vascularized, both benign and malignant tumor. So because they need nutrient, they need oxygen, they need many things, and they need blood supply. Like normal tissue, tumor require delivery of oxygen and nutrient and removal of waste products. Cancer cells and also large benign tumors can stimulate neoangiogenesis, during which new vessels spring from previously existing capillaries. But tumor vasculature is abnormal. The vessels are leaky, the vessels are irregular, they are dilated, and they show an irregular pattern of connection. Angiogenesis is required not only for continued tumor growth and also for access to the vasculature and for metastasis. We said that finally the tumoral cells prefer to spread through the body, okay? And for this spreading and for this metastasis, they need vessels. So the tumoral cells need vasculature for nutrient, for oxygen, and also for spreading and for metastasis. Neovascularization has dual effect on tumor growth. Perfusion supply needed nutrient and oxygen, and also the newly formed endothelial cells. Endothelial cells are the cells inside the vessels. The cells, we call them endothelial cells. These endothelial cells can stimulate growth of adjacent tumoral cells by secreting some growth factors. Okay, so these newly formed endothelial cells can secrete some growth factors and can help to proliferation of the tumoral cells more and more. Angiogenesis is necessary biologic correlate of neoplasia both in benign and malignant. Early in the growth of tumor, most human tumor don't induce angiogenesis in the first stage of the tumor. And they remain small or in situ for many years until the angiogenic switch terminates this stage of vascular quiescence. So at first, many tumors are small and, or sometimes we call them in situ. When they are in situ, they show no invasion and no metastasis. And if we find them in this state, we can cure them only by removing of this area.
Surgical.
Yes, certainly. But after metastasis and after many invasion to the surrounding tissue, the curing is very hard.
Angiogenic.
Yes, after angiogenic stage. After angiogenic stage, this is very hard because the tumoral cells start to spreading through the body and they enter to the blood
Having
vessels.
access to the
Yes, they have
circulatory
access.
system.
Yes, they have access to any part of the body by the circulation of the blood. Tumor angiogenesis, as I said before, like anything, is controlled by some pro-angiogenic factors and some anti-angiogenic factors. The molecular basis of angiogenic switch involves increased production of angiogenic factors, and on the other hand, loss of angiogenesis inhibitor. So as I said in proliferation, I said we have some genes for that they are proto-oncogenes. We have activation in this area, and we have some area for tumor suppressing, and we have inactivation of this area. This is like that. We have activation in pro-angiogenic factors and inactivation of anti-angiogenic factors. So we have, finally, we have many vessels and we have many vasculature and many connection of blood vessels in the site of tumor. These factors are produced by tumoral cells, by inflammatory cells in the background of tumor, and by the stromal cells or tumor-associated fibroblasts. You know,
Do you mean that we can inhibit the angiogenesis, like by some drugs or something else?
yes, exactly. As I said before, we have some targeted therapy for each of these systems. Okay. Sometimes we can use from these drugs. Sometimes we have some drugs like this. What?
Also the radiotherapy?
No, radiotherapy acts in other way. Yes. But sometimes we have some surgery way for blocking the, for example, the vessel which is supply the tumor. Sometimes we can send some emboli-like material, some drug to that area, and we can stop the blood supply to tumor. And also we have some drugs, for example, for inhibiting the activity of pro-angiogenic factors.
So if we initiate this procedure by blocking the, the blood vessels of the tumor, why you don't remove it at all and, get rid of it?
Okay, sometimes, for example, sometimes the tumor is so large at first, okay, and the tumor is attached to the surrounding tissue. And sometimes the surrounding tissue are so critical. For example, some critical vessels, the tumor is near that. Okay, and by this way we can at first give some drugs for shrinkage of the tumor. This tumor becomes smaller, and after that the removing of a tumor is very easier. Yes, exactly. Okay. In any tumor, we have some tumoral cells, for example, like this. You know that tumoral cells show pleomorphism. What do we mean by pleomorphism?
Different shape and size.
Different shape and size, yes. For example, if the normal cells is like this, for example,
Yeah, irregular.
regular and monomorph and like each other. But tumoral cells, for example, yes, different shape, different size of and irregularity in the nuclear border, and sometimes some prominent nucleoli, and for example, like this, we have many mitotic figure, and sometimes we have abnormal mitotic figure. You know about abnormal mitotic figure?
No.
Yes. What's the meaning of abnormal mitotic figure or abnormal mitosis?
There's abnormal cell deviation.
Yes, in abnormal cell deviation. For example, in normal situation, we see mitosis like this. Okay, like this. Because the chromosomes are here as a line in the middle part of the cell. This is a normal mitosis. But in abnormal tumoral cells, for example, some chromosomes are here, some of them are here, and we have some bizarre shape of.
You mean after deviation, one of the new cells gonna be smaller, too smaller, and the other one is gonna be too larger than the other?
Yes, exactly. In the tumor area, we have some tumor parenchyma. This is tumor parenchyma. And also in the background, we have some other cells like inflammatory cells, like macrophages, some lymphocytes. This is microenvironment of the tumor, okay? And this microenvironment now is very important because we know that They are some way like immunotherapy, and we can use from some cells like these lymphocytes for killing the tumoral cells, and this is immunotherapy. This is a new way for curing these patients. Okay, so we have some lymphocytes, some macrophages like this, and also in the background we have some stromal cells or fibroblasts, some spindle-shaped cells in the background. In the background of any tumor, we have some cells like this. Okay, these cells are very important for producing growth factors, producing pro-angiogenic factors, and they can help to the proliferation of the tumor parenchyma. Okay, you know that many enzymes and many proteins are important in this process. Some proteases are involved in regulating the balance between angiogenic and anti-angiogenic factors. They are elaborated directly by tumoral cells and also by the stromal cells by stimulation of the tumoral cells. Many proteases can release pro-angiogenic basis fibroblast growth factors that are stored in the extracellular matrix. Conversely, the angiogenesis inhibitor like angiostatin or endostatin, this statin means stopping an inhibition, are produced by proteolytic cleavage of plasminogen and collagen respectively. It means that by cleavage of angiostatin we have plasminogen, and by cleavage of endostatin we have
Collagen.
yes. The angiogenic switch is also controlled by several physiological stimuli like hypoxia. In any situation of hypoxia, we have proliferation of blood vessels for blood supply and oxygen supply.
That's to inhibit the growth of the tumor?
Yes. Hypoxia want to inhibit the growth, and by vasculature and making some vessels, they want to proliferate. The tumoral cells want to give more oxygen and proliferate. Related lack of oxygen due to hypoxia stabilize some factors. They are not important. And yes, finally, we have some other cytokines and pro-angiogenic cytokines like VEGF or vascular growth factors. This factor creates an angiogenic gradient that stimulates proliferation of the endothelial cells. And finally, by the proliferation of endothelial cells, we have some new vessels toward the tumoral cells. Mutations involving tumor suppressors and oncogenes in cancer also tilt the balance in favor of angiogenesis. For example, we said P53 is very important mutation in the tumors. P53 normal situation stimulate expression of anti-angiogenic molecules, okay, like thrombospondin 1, and also repress expression of pro-angiogenic molecules such as VEGF. So, loss of P53 in the mutated cells, by loss of P53, we have a permissive environment for angiogenesis. Development of therapeutic agent that block angiogenesis. A cardinal example is bevacizumab, is a new drug, a monoclonal antibody that can neutralize the VEGF, can neutralize the activity of this factor, and this drug is approved for use in treatment of many types of cancer. Now about ability to invade and metastasis. Metastatic cascade can be subdivided into two phases. At first, we have invasion to the extracellular matrix, and after that, we have vascular dissemination and homing of the tumoral cells. You know about the basement membrane and also interstitial connective tissue. You know that there are many types of proteins and many types of collagen in basement membrane. So tumoral cells need to make some enzyme for cleaving these sites and for making some way for their invasion and for the access to the blood vessels and metastasis. Tumoral cells must interact with the extracellular matrix at several stages in the metastatic cascade. At first, they must reach the underlying basement membrane. After that, they traverse the interstitial connective tissue. And finally, they gain access to the circulation by penetrating the vascular basement membrane. And you know, this cycle is repeated more and more, and the tumoral cell emboli extravasate at a distant site. Okay, you have 3-minute time for this slide.
Is it possible to zoom in?
Really?
Form cancer cells, the first cell. And then there is the formation of primary tumors by the multiplication of that cancer cell and further mitosis and divisions. So after the multiplication, that colony of the cells need blood supply and their nutrients, so they do angiogenesis, the formation of new vessels, and after as she mentioned that angiogenesis is for two purposes, first for the nutrition and second for the separate. Now the cancer will separate by the vessels, and how it happens, first it will close the basement membrane, it will attach to the basement membrane and invade it by the cleavage factors, and then it will invade, then it will cleave the collagen in the ECM. After that it will attach to the vessel membrane and enters the bloodstream, and it will attach to the blood cells, lymphoid cells of the humans, and
Why they attach to the?
so for the easy separate.
Yes, for treating the immune system.
Mr.
They,
Dave.
they hide, yes, they hide behind the platelet
It
and
will attach to the lymphoid cell, and then the tumor emboli will move to its destination, and where when it will reach the destination, another point where there it will again attach to the basement membrane and cross the collagen, and the same mechanism it will come out and start colonizing there and form the secondary tumor at that site. Yeah, and the
Very
cycle will start again.
very good. Thank you so much. Yes, you know, at first we have a transformed cell, a malignant cell, okay? We have the malignant cells and primary tumor here. When tumor is only in the site above the basement membrane, we call that in situ tumor, okay? So at first, for example, this is a basement membrane, for example, epithelial tumor, like squamous cell carcinoma. They are primary tumors of the skin, and the skin is a type of epithelial tissue. Okay, in the epithelial tissue we have some epithelial cells like this. This is the normal cells. Because of some DNA damage, for example, in the skin, because of UV damage, we have a transformed mutated cells. For example, this cells is mutated, and after that, by any advantages and features which I said and discussed about them, these cells start to proliferate, and we have clonal expansion growth of these cells. What's the meaning of clonal? You know about monoclonality and polyclonality.
Many cells will form from the single cell.
A single cells, yeah, with same features and same characters. They are clonal expansion or monoclonal expansion. This is very important, the difference between monoclonality and polyclonality. For example, sometimes we have proliferation of cells, but these cells are polyclonal, like infectious disease. In the infection, we have proliferation of the immune cells like lymphocytes and plasma cells, but they are polyclonal and they are from different antigen, and some of them have some antigen and some don't. But in the tumors, we have monoclonal expansion or clonal expansion. So these cells start to proliferate like this. And when the tumor are limited by the basement membrane, when they are limited by these, we call them in situ. Okay, we call them in situ tumor or in situ malignancy. In this situation, they have no access to the blood vessels, they have no invasion, and this is the importance of a screening way. By a screening way, we can find the tumor in this stage and in this step. Okay. So at first they are in situ, but after that, by some other mutation and by making some blood vessels, they start to invasion to the basement membrane, okay? At first, they reach here and they are into the extracellular matrix like here, and after that, they can travel in this area and they can enter to the blood supply. And in the blood supply, sometimes they make some emboli, some aggregation, some small aggregation like emboli, and they can attach to some immune cells, some lymphocytes, and some platelets. And finally, by the same mechanism and by same enzymes and proteases, they can make some pores here in the basement membrane of the blood vessels, and they can go to the extravasate from the blood vessels and go to the another part which is good for proliferation of tumor. Evasion of the extracellular matrix is an active process that requires several sequential steps. At first, we have loosening of intercellular connection between tumoral cells. Then we have local degradation of basement membrane and interstitium. And then we have changing attachment of tumor cells to the extracellular matrix proteins. And finally, we have locomotion. At first, about loosening of tumoral cells. Loosening of tumoral cells is the first step in the metastatic cascade. The important protein and molecule in this step is E-cadherin. What is E-cadherin? What? Yes. This is like a glue, and in the epithelial cells, they can keep the cells together. So in the epithelial tumors, we have loss of normal function of E-cadherin. Because of submutation, the function of E-cadherin is lost. In this picture, you can see what are these black line? E-cadherin. We said that these are like glue between epithelial cells. This is a mutated. Tumoral cells. You can see that we have no attachment to the surrounding epithelial tissue, and we have loosening of intercellular junction. After that, we have local degradation of basement membrane and interstitium. Tumoral cells secrete some proteolytic enzymes themselves, and also some stromal cells. As I said before, in the background of tumor, we have some stromal cells, and also we have some inflammatory cells that they can produce some proteases. Multiple different family of proteases like MMP or matrix metalloproteins and other protein and molecules are important, and finally they are implicated in tumor cell invasion. In this picture, you can see some types of collagenase, for example, type 4 collagenase, because type 4 collagen is one of the most important protein in the basement membrane. So for cleaving the basement membrane, we need some proteases and some type 4 collagenases. The tumoral cells themselves and also some inflammatory cells and other stromal cells start making some proteases. And after that, we have change in attachment of tumoral cells to the extracellular matrix proteins. You know that the normal epithelial cells have some receptors such as integrin to the basement membrane. To the basement membrane laminin. And collagen that are polarized at their basal surface. This receptor helps to maintain the cells in the resting and differentiated state. So lots of adhesion in normal cells, leads to induction of apoptosis. But we know that tumoral cells are resistant to apoptosis. Finally, we have locomotion or propelling tumoral cells through the grated basement membrane and zone of matrix proteolysis. And such movement seems to be potentiated by tumor cell-derived cytokines such as autocrine motility factors. And you can see here the migration of tumoral cells through the basement membrane. Finally, we have vascular dissemination and homing of tumoral cells. When the tumoral cells enter to blood, some of them are vulnerable to destruction by host immune cells, but some of them can escape from immune system by adhering to the leukocytes and
platelets.
Yes. Extravasation of tumoral cells or emboli involve at first adhesion to the vascular endothelium, and after that egress through the basement membrane of the vessels into the organ parenchyma by the mechanism similar to those involving the invasion. Okay, now about site of extravasation and organ distribution of metastases. For example, the distribution of metastasis site is different between the tumors. For example, in breast cancer, the first area of metastasis is axillary lymph node. Is axillary lymph node. Why? Because of the normal lymphatic drainage of the breast. We should know about the lymphatic drainage and the blood vessels of tumor, and by that we can find the first area of metastasis and the first lymph node which is involved by tumor. The first lymph node which is involved by tumor, we call them sentinel lymph node, yes. And finding the sentinel lymph node is very important because sometimes in the surgery, the surgeon needs to find the sentinel lymph node for removing the involved lymph node. So, the site of extravasation and site of metastasis is related to the location of primary tumor and also the vascular or lymphatic drainage of that area. Many tumors arrest in the first capillary bed they enter. More frequent metastasis is in lung and liver. Why in liver?
Because venous return always goes to the liver.
Yes, exactly. The first capillary bed in in this system is liver and lung.
All cardiac output goes to lung
Yes.
first.
Yes, thank you. Sometimes, in many cases, the natural pathway of drainage can't readily explain the distribution of the metastasis. For example, as I said before, in prostatic carcinoma, the bone is involved, and we can't explain this by the drainage and connection of the blood vessels. And some tumors, like lung cancer, tend to involve the adrenal gland. And also they almost never spread to the skeletal muscle. So we have a term organ trophies in tumor. Some tumors have some organ trophies, and this is maybe because of adhesion of some molecules and some chemokines, and also chemokine receptor that their ligand are expressed in the endothelial cells in that metastatic site. Okay. For example, a tumor make an antigen or a chemokine, a protein, which that protein's receptor, for example, is only in adrenal, and because of that, this tumor in metastasis prefer adrenal gland. And also we know, for example, that receptor is not in skeletal muscle, so this tumor don't metastasis to the skeletal muscles in any situation.
Could you please repeat? Could you please repeat why it's
About organ
adrenal?
tropism.
No, about the adrenal gland. Why it's
This is as an example. We said here that, for example, lung carcinoma prefer adrenal gland.
why?
In practice, we see this. Okay, in practice, we see this that many patients with lung carcinoma show metastasis in the adrenal, and they don't know why at first. And now we know that maybe some cytokines and some proteins, some molecules which is produced by lung cancer, these receptors are in the adrenal. Okay, so because of that receptors, this tumor prefer to go to that area. This is organ trophies in tumor. Okay. Another feature of the tumoral cells, you know, is evasion of the immune system. Most tumor arise in immunocompetent host, and accordingly, a likely strategy for sepsis is to trick the immune system in such a way that the tumor fails to be recognized by the immune system. And I think you learned in the chapter of immunology about this immune system. Okay, now about enabler of malignancy. You know that in genomic instability that we said we have genomic mutation and genomic alteration in the tumoral cells. The genomic instability occurs when both copies of the gene are lost. Okay, and also we have tumor promoting inflammation. Inflammation is very important because we have many inflammatory diseases that they are not malignant, but they can make some environment, some permissive environment for making some malignancy in the background of inflammatory process. For example, a persistent chronic inflammation in response to microbial infections, or as a part of autoimmune reaction, you know about ulcerative colitis, for example, a type of autoimmune disease, and also Barrett esophagus. You know, Barrett esophagus is a condition that we have persistent chronic inflammation in lower part of the esophagus because of the acid regurgitation. And also, sometimes inflammation occurs in response to tumor. Okay, this is, on one hand, we have a tumor because of inflammation, and sometimes we have inflammation after the tumor make. Now about genomic instability. Also, humans are averse in environmental agents that are mutagenic, like many chemicals, radiation, and also sunlight. But you know, cancers are rare outcome of this condition. This state of affairs results from ability of normal cells to repair the DNA damage. The normal cells, the normal people have many mechanisms for repairing the damaged DNA. DNA repair is necessary in maintaining the integrity of the genome. Some persons born with inherited defects in DNA repair proteins are at greatly increased risk for development of cancer because they have damage in one allele, and sometime by other damage, maybe the other allele is damaged again, and after that they show, after that the both copy are lost, and then we have malignancy. Defects in 3 types of DNA repair system are determined. Some of them act in mismatch repair, some of them act in nucleotide excision repair, and recombination repair. A good example for mismatch repair gene is HNPCC. HNPCC is hereditary non-polyposis colon carcinoma syndrome. These patients are some young patients with a malignant tumor in their colon. They have no other polyposis in their colon. They are younger, and their tumor are in the right colon, right side of colon, for example, in secum and in the ascending colon. And also in the pathology examination, for example, they have some morphologic feature like mucinous adenocarcinoma. When a strand of DNA is being repaired, the gene acts as a spell checker. We have some spell checkers. For example, if there is an erroneous pairing of G with T rather than the normal A with T, from the genetic, yes. The mismatch repair genes correct this defect. So we have some genes that act as mismatch repair genes. Without these proofreaders, errors accumulate at an increased rate, and we have finally a mutator phenotype. A characteristic finding in the genome of patients with defect in mismatch repair is microsatellite instability. Okay, now about microsatellites. Microsatellites are some repeats of 1 to 6 nucleotides found through the genome. In normal people, the length of this microsatellite remains constant by any dividing. Okay. But in patients with HNPCC, these satellites are unstable and increase or decrease in length. Who can explain about microsatellite and microsatellite instability? As a review of these things I said. Microsatellites, I said, are, who can explain?
He can change the genome. شنو صارت؟ bonding.
Microsatellite instability, yes. I said that in the genome, for example, like this, we have some area, microsatellite, we call them microsatellite. Microsatellite are some area, for example, from 1 to 6 nucleotides, okay? Like this, for example, A, T, T, C, G.
Keeps repeating.
Okay,
Yes.
like this. We have many, this sequence through the genome, okay? And the length of these is constant by any dividing and in anywhere, okay? But in patient with damage in mismatch repair genes or MMR, by any dividing we have decrease and or increase in the length of this area. And now we have some molecular way for finding the length of this area. And we have some tests for microsatellite instability. We can use the test of microsatellite instability for find the length of this area. For example, in normal people, we have a report of MSI like this. Okay, it means that the length of this area is constant in any cells. But in patient with microsatellite instability, the report is like this. Okay, like this. And in this patient, we can find the colorectal malignancy and cancers very soon in younger patients. In this syndrome, this is a familial carcinoma, for example, sometimes some brothers and sisters, for example, 3 patients in a family, they show this type of malignancy. In this syndrome, colon, in the secum and proximal colon, as I said before, in right side of colon, this tumor happens. And mutations in at least 4 mismatch repair genes have been found to underline. We have some types of mismatch repair genes, for example, MSH or PMS. They have a different name, and this is not important for you. And another disease that some patients carcinoma of the skin in very young age. You know that some tumor normally is seen in the older patient. For example, squamous cell carcinoma of the skin mainly seen in the older patient, for example, more than 60 years. But some patients are childs, but because of this syndrome, we call this xeroderma pigmentosome. Because of that, because they have some types of damage in the DNA repair system, they show these types of tumors very soon and in the childhood. UV rays in sunlight skin can cause cross-linking of pyrimidine residue and prevent the normal DNA replication. This DNA damage is repaired by a system we call that nucleotide excision repair system. But increased risk for the development of cancer of sun-exposed skin is present in this patient with xeroderma pigmentosome. Okay, now about inflammation. This part is very, very important. Okay, as I said before, inflammation can enable malignancy in 2 different settings. At first, persistent chronic inflammation in response to microbial infection or as a part of an autoimmune infection. The increased risk of cancer in patients affected by a variety of chronic inflammatory disease of the gastrointestinal tract, like Barrett esophagus, ulcerative colitis, H. pylori gastritis, hepatitis B, C, and also chronic pancreatitis. These situations are common situations that after a chronic inflammation, we can see some types of malignancy. As with any cause of chronic tissue injury, there is a compensatory proliferation of cells in an attempt to repair the damage. So for repairing the damage, we have compensatory proliferation of the cells. This regenerative process is aided and abetted by a plethora of growth factors, cytokines, chemokines, and other bioactive substances produced by activated immune cells collected at the site. Persistent cell replication and reduced apoptosis under this condition place the cell at risk of acquiring some other mutation in one or more of the cancer genes. In addition, inflammatory cells such as neutrophils can contribute to carcinogenesis by secretion of some ROS and some DNA damage. And sometimes we have inflammation in response to the tumors. Inflammatory reaction is protective since it represents an attempt by host to destroy the cells. And inflammation leads to tumor promoting activity by producing growth factors and inflicting additional DNA damage. So, in one hand, they can help to proliferation of tumoral cells, and in the other hand, they can destroy the tumoral cells. And they have different activity, the inflammatory cells. The inflammation often thought of as a protective response against tumor, and also can paradoxically enable malignancy. So we call that this is double-edged sword. So in the some inflammatory disease, for example, in ulcerative colitis. In the ulcerative colitis, we have an environment. So in the site of inflammation, break. Okay, 5 minutes. Okay, only 5.